Welcome back to the lab. We've learned how to manipulate genes, clone cells, and verify our results. But what if we want to read the code of life itself? Today we're diving into the revolutionary technique that made this possible: DNA sequencing. We'll go from the classic methods to the modern technologies that are transforming medicine and research. And we're back. At its core, DNA sequencing is the process of determining the precise order of nucleotides within a DNA molecule. Think of it as reading the instruction manual for an organism. This fundamental technique allows us to understand the genetic code for research, diagnostics, and even personalized medicine. It's the ultimate tool for confirming the genetic identity of our cloned genes, a topic we covered in a previous episode. The process has evolved dramatically over time, starting from manual, low-throughput approaches to today's highly automated, high-throughput technologies. The basic workflow involves 1. Sample preparation. We start by extracting the DNA, and often we need to fragment it into smaller pieces for easier analysis. We might even use PCR to amplify the gene of interest before we get to the sequencing part, a technique we've already covered in detail. 2. Sequencing. This is where we run the reaction to generate the DNA sequence. 3. Data analysis. Finally, we use powerful computational tools to assemble the sequence and identify any errors or mutations. Let's take a look at the key sequencing methods that have shaped modern biology. Sanger sequencing. This is the classic method. It uses enzymatic reactions to generate DNA fragments of varying lengths, and then we separate these fragments using electrophoresis. We've covered gel electrophoresis in a previous episode, and it's this same principle that allows us to read the DNA sequence. This method is still widely used today for its accuracy, especially for sequencing single genes or for validation. Next-generation sequencing, NGS. This is the powerhouse of modern sequencing. NGS technologies allow for the massively parallel sequencing of millions of DNA fragments at the same time. This dramatically increases the speed and throughput, making it perfect for sequencing entire genomes, a process that would be impossible with traditional Sanger sequencing. There are several types of NGS, including pyrosequencing and single-molecule sequencing, all designed to read genetic information in real time. These technological leaps have made sequencing faster, more reliable, and much more accessible. Like any advanced lab technique, things can go wrong, but a good scientist knows how to troubleshoot. Problem 1. Poor DNA quality. Solution: low-quality DNA can lead to low-quality reads. Always assess your DNA sample's concentration and integrity before starting, as these are critical to a successful run. Problem 2. Low number of recovered reads. Solution: the amount of DNA and the number of PCR cycles during your library preparation can significantly affect your results. Make sure you optimize these parameters. Problem 3. Contamination or low complexity. Solution: screening your library quality with tools like FAQCs can help detect issues like adapter contamination before you commit to a full sequencing run. By paying close attention to your sample preparation and quality control, you can ensure reliable and accurate sequencing results. So why go to all this effort? The applications of DNA sequencing are truly transformative for human health. Personalized medicine. Sequencing can identify genetic mutations that make a person more susceptible to certain diseases or that respond better to specific treatments, especially in cancer. This allows doctors to create targeted therapies that are more effective and have fewer side effects. Epigenetic analysis. New sequencing methods can even detect epigenetic changes alongside genetic ones, which improves diagnostic accuracy. Transcriptomics. RNA sequencing allows us to assess changes in gene expression, which helps guide therapeutic decisions. DNA sequencing is no longer just a research tool, it's a key part of clinical diagnostics and personalized medicine. From the manual gels of Sanger sequencing to the massive data output of NGS, DNA sequencing has evolved to become a cornerstone of modern biology and medicine. It is the ultimate tool for reading and understanding the blueprint of life. If you're fascinated by genetics and the incredible tools that make this research possible, hit that subscribe button and ring the notification bell. Our How to Biotech playlist is your go-to resource for understanding these complex techniques. Join us next time as we explore another exciting frontier in the world of biotechnology.